My name is Janos Filep. I'm from the University of Montreal. I have made a presentation for the 21st Congress on Heart Disease on CpG DNA regulation of coagulation and its implication for acute coronary syndrome. So that CpG DNA commonly refers to a DNA which may be released from bacteria or mitochondria, and the CpG, that is a common structural feature of this DNA, that in this motif the cytosine is not methylated as compared to eukaryotic DNA when cytosine is methylated, and that for that reason it is not recognized by the immune system. So the question was that CpG DNA can cause inflammation in many different model systems. So it is a question what we asked, that is whether it could also affect coagulation. So that we have used cultured human coronary artery endothelial cells and mice to address these issues. In cultured endothelial cells, CpG DNA induced a time and dose-dependent expression of tissue factor, which is a main inducer of coagulation in the heart, as well as in many other organs. That it enhanced protein transcription, a protein expression, so this enhanced protein content inside the cells, as well as the secreted proteins. But perhaps more importantly, it also induced activated tissue factor expression on the surface of endothelial cells. By contrast, that CpG DNA decreased tissue factor pathway inhibitor, a naturally occurring inhibitory protein which block tissue factor activity and thereby countering coagulation. It's once again, this effect was at the transcriptional level. It appeared to be a time and concentration dependent. That this action of CpG DNA was mediated through toll-like receptor 9, as evidenced by inhibition of toll-like receptor 9 signaling through an inhibitory oligonucleotide or transient downregulation of TLR9 expression by siRNA technology. So in the mouse, that when we injected CpG DNA, that it shortened bleeding time and enhanced the amount of circulating thrombin antithrombin complexes, that those are indirect measures of coagulation. It's once again that this effect was time and concentration dependent. A genetic dilation of toll-like receptor 9 resulted in a complete loss to responses to CpG DNA. When we inhibited a tissue factor by an antibody before injection of CpG DNA, this also rendered mice unresponsive to CpG DNA, indicating that the effect was due to coagulation and not other component of hemostasis. And the last piece of evidence, and perhaps the most intriguing, that is linking CpG DNA to the coronary circulation, is that following intravenous injection, that we detected enhanced tissue factor expression in coronary artery endothelial cells in this mice. So that taken together, that this data suggests that activation of toll-like receptor 9 by either bacterial DNA released from bacteria or mitochondrial DNA following tissue injury links inflammation to the coagulation, and this requires toll-like receptor 9 activation, downstream signaling like NF-κB activation, and a shift between the balance of procoagulant and anticoagulant factors like upregulation of tissue factor expression and downregulation of tissue factor pathway inhibitor. This data also raised the possibility that targeting toll-like receptor 9 signaling, for example by inhibitory oligonucleotide, might be a novel therapeutic approach to counter the inflammation and thrombotic consequences of conditions when CpG DNA are abundantly expressed, such as in myocardial infarction and other diseases.